Hello, everyone. My name is Sen Yan, a PhD student from Southeast University in China, and my supervisor is Professor Ning Gu. Next, I will briefly introduce my work. My work is focused on the use of magnetic materials in cell therapy. My presentation is entitled Extracellular Magnetic Labeling of mesenchymal stem cell spheroids with ferumoxytol for MIR checking. It's a new cell labeling method using ferumoxytol. My presentation revolves around 4 parts: background, research results, conclusions, and acknowledgements. First, I will introduce the background of our research. Mesenchymal stem cell, MSC, have been used for the treatment of various diseases because they have many functions such as homing, anti-inflammatory, but the development of MSC therapy is culturally challenged by the loss of the transplanted MSC and the failure of function due to the high apoptosis after transplantation. In order to overcome these difficulties, MSC 3-dimensional culture technology, we call MSC spheroids, can significantly improve different MSC functions due to cell-to extracellular matrix interactions and cell-to-cell interactions. Currently, there are 2 main types of cell spheroid preparation methods. The first one is the scaffold-free method. It's based on the suspension of cells. The second one is the scale-based method, like hydrogels. Now, this method are the main way to prepare spheroids because it can be mimic the microenvironment of the organism. MSC spheroids will better therapeutics efforts and have been used in various preclinical animal studies, such as vascular regeneration, colitis, spinal cord injury, and stroke for localized lesion transplantation. During the development of MSC spheroid applications, it is important to understand whether the spheroids are successfully transplanted to the site and how long the spheroids can function after transplantation. Bioimaging can be a good way to observe spheroids, and MIR checking has the advantages of safe and non-invasive. MIR checking needs 3 elements: MIR, cell labeling materials, and suitable labeling method. Currently, the traditional approach is to use the uptake of IRO-based nanoparticles by cells to enhance the construct of cell. To ensure the safety of labeled cells, medical IRO-based nanoparticles have the advantages of clinical translation. So ferumoxytol is an FDA-approved nanoparticle and have been used to MIR construct agent, cell labeling, cell therapy, and drug delivery. In China, a product has been licensed for clinical study called Return. Many studies have demonstrated the cell labeling efficiency of ferumoxytol is very low, and the cell labeling with ferumoxytol usually requires high labeling concentrations and the use of toxic transfected reagents such as lysine and protamine sulfate to promote uptake of MSC. This will limit its clinical application. So we want to develop an easier, safer, and efficient method for spheroid labeling. So based on our former research, we want to use a mimetic hydrogel as a metric material for MSC sphere formation, and add ferumoxytol directly during the preparation of spheres for labeling. Next, we designed two types of germa PGDA hydrogels for culture MSC by adjusting the ratio of germa to PGDA. These two types of hydrogel have different stress relaxation time and Young's modulus. And MSC seeded on the two types of hydrogels showed different morphologies. These PGDA germa hydrogels provide a suitable microenvironment for MSC sphere formation. Then, during cell culture, we added ferumoxytol in medium. After culture for 1 day, spheres gradually formed dense and completed structures and screened a large amount of ECM proteins. After 3 days, the spheres showed an average size of 117 micrometer. By pollen stem, we found the blue aryl singles into the spheres. This result confirmed MSC spheres can be labbed with ferumoxytol without transfection reagents. In addition, we also modulated the different size of spheres, showing the spheres of different size could be labbed. To improve the labeling method is extracellular labeling. We used the TEM to observe the location of ferumoxytol in spheroid. In this figure, we cannot find accumulated ferumoxytol within the cytoplasm of cells in spheroids and the traditional cell culture cells, while aggregates of ferumoxytol was found in extracellular space. This result indicates that our labeling method is independent of cellular uptake of ferumoxytol. It relies on accumulation of ferumoxytol in the extracellular space. Ferumoxytol was localized in the extracellular matrix of the spheres instead of the cytoplasm. Using this method, we changed the incubation concentration of ferumoxytol and adjusted the size of the spheres. Staining and the aryl quantification method, ICP, showed the aryl content of spheres can be adjusted by control the concentration of ferumoxytol and the size of the spheres. Higher concentration and the large size of spheroid can load more ferumoxytol. In addition, we continued to culture spheres after labeling for 14 days. The elution amount of ferumoxytol from spheres was measured, and the results showed ferumoxytol was maintained inside the spheres. According to these results, we confirmed our extracellular magnetic labeling method can lab the spheres, and this labeling method is stable and durable. The biosafety of this method and function of spheres also need to be measured. We used some experiments confirmed the use of ferumoxytol is safe and no side effects of incubation on the in vitro and in vivo activity of MSC spheres. Also, this labeling method does not affect the cell phenotype, stemness, and function, and it's suitable for transplantation and repair. Then we performed the in vitro MIR of magnetic spheres. In this figure, we can see obvious dark T2 MI singles, and the intensity is influenced by the concentration of ferumoxytol. So we transplanted magnetic spheroids into mouse for different days MIR tracking and showed T2 signals maintained up to 7 days. Compared to the histological result, transplanted magnetic spheroids cannot be observed about 10 days. These results confirmed the MIR signals can reflect the retention of spheroids. Above all, I introduced my work on extracellular magnetic labeling of spheroids. This method is easy to operate by using the properties of MSC spheroids to lab ferumoxytol into the inside of the spheroids. Ferumoxytol do not enter into the MSC and have less toxicity and impact on stem cell. This is more conducive to clinical translational applications. The hydrogel mimics the in vivo microenvironment and provides better condition for improving cell function. Spheroids of different size and the labeling amounts can be prepared, and the labeling amounts can be concentration controlled to meet different needs. Magnetic MSC spheroids can be used for MRI imaging after in vivo transplantation to determine the success of MSC spheroids transplantation, and to track the duration of spheroids function in vivo. It can provide more information for transplantation therapy of MSC spheroids. Finally, I want to thank my supervisor and some teachers and students in our lab. Thank you all for listening. Thank you.
Thank you very much, Sen Yan. Anyone has questions? So I have one. So does ferumoxytol have a coating to ensure the uptake?
The ferumoxytol is coating with the PSC. When we use the ferumoxytol, we do not coating any materials.
Okay, and thank you. And
we can use the ferumoxytol directly. It's a simple way.
Okay, thank you. Another question.
I got a layman question. First, thanks, Sen Yan, for the interesting presentation. So, in your work, is there any optimal size for the magnetic spheroids?
Hey, sorry, I didn't catch all of that question. Please pardon?
So during the labeling, is there any optimal size for the spheroids?
When labeling, I found no side effect.
Okay, great, thanks.
Okay, you're welcome.
Sorry, another question. I have one, but I don't want to. Okay, so I'll ask it. When you mention the hydrogel, how do you choose the hydrogel? I mean, which properties are you trying to get from it?
When preparing hydrogels, I designed two types, and in these two type hydrogels, MSC MSC showed different velocity, and we controlled the Young's modulus and stress relaxation time, and we choose the suitable elements for cell for formation multi-cell spheroids. Yeah.
Okay, we have one last question in the chat, but it has to be really quick because we have only 1 minute. So Michael asked, if developed as a medicine, would you keep the magnetic tracking or would it be unnecessary?
Okay. I think this cannot affect the outcome of the cell therapy, and if the singles is disappear, I will thought the spheroids is missing, and we can use this thought and to control the spheroid is not on the side of the transplantation side.
Okay, thank you. So thank you, Sen Yan,